And without further ado, I would like to hand over to the first speaker and wish you an interesting session on AI in retinal diseases.
Hello, everybody. I am Professor Shobha Shivaprasad from Moorfields Eye Hospital. Today, my topic is on detection of diabetic retinopathy from ultra-wide field scanning laser ophthalmoscopy. These are my financial disclosures. Deep learning-based technologies for diabetic retinopathy detection has been well established using conventional fundus photographs. For the past 5 years, we have seen significant number of publications on development and validation of these algorithms. However, they have all been done on conventional retinal fundus photography. They also show a very high accuracy. These are examples where the accuracy have gone up to 0.95 or above. This smaller paper looked at the accuracy of ultra-wide field fundus ophthalmoscopy in detecting proliferative diabetic retinopathy. Ultra-wide field fundus cameras are now widely used, and there is significance in its importance in terms of grading diabetic retinopathy, as peripheral changes seems to be important in predicting disease progression. In this study, it was a multi-center collaborative study, and we aimed to look at developing a new deep learning system for detection of diabetic retinopathy lesions within the 7-field fundus photography of the ultra-wide field scanning laser ophthalmoscopy. And I'll explain that a little better. The two aims were to look at automatic assessment of gradability of ultra-wide field images as to whether they were gradable or ungradable, and then to go on to detect referable diabetic retinopathy and vision-threatening diabetic retinopathy on these wide field retinal images. So as you know, all the diabetic retinopathy screening severity scales have been developed from standard 7-field ETDRS grid. So what we did was we superimposed this 7-field ETDRS grid onto the ultra-wide field SLO images to look for gradability within that 7-field. So ungradable images were classified as any artifacts that were present within the standard 7-field ETDRS, or if the image was blurry because of media opacities or the image had a low luminance. Below, you can see the shell that we used to define that area. The next part of the study was to look at ground truth labeling of referable diabetic retinopathy and vision-threatening diabetic retinopathy using the International Clinical Diabetic Retinopathy Severity Scale. Referable diabetic retinopathy was defined as any DR severity level of moderate NPDR or worse, and or presence of diabetic macular edema, while vision-threatening diabetic retinopathy was severe NPDR, PDR, and or presence of diabetic macular edema. You can see that there were 9,392 ultra-wide field images from 1,903 eyes from 1,022 subjects. Now, there were 5 centers involved in the study. The study was led by Carol Cheung and Fang Yao Tang from Chinese University of Hong Kong. And we had 4 other centers, one from UK, 2 from India, and another one from Buenos Aires in Argentina. And you can see from this table that there was no difference in terms of demography, duration of diabetes, or type of diabetes between the gradable and ungradable images in any of these centers. We then looked at the comparison between non-referable diabetic retinopathy and referable or vision-threatening diabetic retinopathy images. Here again, there were no differences in terms of age, gender, duration of diabetes, or type of diabetes between these images. We then progressed to look at the gradability assessment. Firstly, the primary cohort was from Hong Kong, and you can see that the AUC was very high at 0.923. It was then looked at the other 4 centers. They had varying AUCs, but every one of them was above 0.8, and the sensitivity and specificity was also high. It varied depending on the centers, but on the whole, the accuracy was very high in terms of gradability and ungradability. Now, coming to the accuracy levels of referable diabetic retinopathy and vision-threatening diabetic retinopathy detection, again, you can see that we did the AUC of all the 5 centers, and all of them had an AUC of 0.905 or above, with good sensitivities and specificities for referable diabetic retinopathy. And for vision-threatening diabetic retinopathy, the AUC was more than 0.910, again with high sensitivity and specificity. And the heat map did show the area where the computer algorithm identified the microvascular changes. So in summary, our proposed deep learning system is able to provide automated image quantity assessment and detection of referable diabetic retinopathy and vision-threatening diabetic retinopathy using ultra-wide field images with excellent performance. This system offers a deep learning-based technology, and this has the potential to be applied in clinical settings to complement and enhance existing diabetic retinopathy screening programs, especially because most of these patients with any form of moderate NPDR onwards are referred into secondary care where ultra-wide field imaging is used immensely. This system will allow us to further make our DR pathways more efficient, save resources, and use our workflow more efficiently. I would like to thank all the collaborators, especially Carol Chung and Feng Yu Tang, who did all the analysis and write-up of the study. Thank you very much.
Hello, my name is Sebastian Waldstein, and I'd like to thank the organizers for inviting me to speak on the current and future status of AI in retina. So if we consider the current status that we are in using AI in our clinical practice, I think momentarily we are seeing mostly the replication of easy but potentially labor-intensive tasks. Those are tasks that we as ophthalmologists can do as routine of our practice, but it just takes a lot of time. So for instance, this includes screening for eye diseases on fundus photographs or on OCT, or also detecting and quantifying biomarkers that we are already aware of on OCT images. I think in the future, we will move towards tackling tasks that are also very difficult for humans. So here, AI has the potential to show superhuman performance. And these tasks include things such as correlating morphology and vision, discovering patterns, clusters, or anomalies in large data sets, and using AI to predict future outcomes and disease trajectories. Let's first focus really quickly on what's already there. And I think ophthalmology is at the forefront, and particularly retina. What you see here is a system called IDXDR, and it consists of a robotic camera that requires only 4 hours of training, and then it takes images of the retina and comes up with a diagnosis of whether the patient has diabetic retinopathy and what grade or no diabetic retinopathy. And although this whole idea is not overly sophisticated, the system works really well, and it was in fact the first ever system in medicine, first ever AI system that was approved by the US Food and Drug Administration. So here it's just a great example that ophthalmology is really at the forefront of things in AI. So what the system does is it detects basically known biomarkers. So after an initial image quality assessment, biomarkers such as hemorrhages, microaneurysms, and so on are detected, and then these biomarkers are used to classify the image into a referable or non-referable diabetic retinopathy or a healthy case. We as retina specialists, we are more interested in really the detail of retinal morphology. And here OCT really gives us a tremendous level of detail that we are currently not really using in our clinical practice, or maybe not quantifying it, because it's just too laborious to manually measure every little fluid pocket. And here AI gives us the opportunity to quantify this fluid very precisely. And it has been shown repeatedly that AI can do this as accurately as human experts, but it's just more reproducible, much faster, and much cheaper. And while we are not using quantified variables such as the volume of fluid in our practice at the moment, I think these algorithms will come to our practice soon, and we will be using them in the future. So to sum up the current status, at the moment, in terms of replicating human tasks, AI is already as accurate as human experts. Some systems are already FDA approved, and others will arrive in your imaging systems soon. These new screening systems will change referral patterns, and automated quantification of retinal pathology may be required, in fact, in the future for therapeutic management. Now let's move on to what I think the future will bring. So in the future, AI will be used to tackle tasks that are also difficult for humans. And these tasks include understanding the link between morphology in the OCT, for instance, and vision, to discover patterns and clusters in data and to predict outcomes. In the first example here, OCT is used to, or deep learning is used to link OCT to microperimetry. And as you know, microperimetry is a very tedious examination method, but it gives a great detailed level of information. And so the idea is to use deep learning to use OCT, which is very quick and reproducible, and find the link between OCT and microperimetry. And this is done in a fully automated way and works really well. So here you see a few example cases of OCTs and the corresponding predicted and ground truth microperimetry map. And you see that they overlap really well. And that, for instance, in this case of geographic atrophy, the central area of atrophy was picked up by the system as an area with low retinal sensitivity, as you might expect, and as you see in the ground truth. Now, if we go beyond that, we can use deep learning and artificial intelligence just to learn patterns in large-scale data. And we did this a couple of years ago, where we took 60,000 OCT volumes of patients with a neovascular AMD, and we just fed them into an autoencoder and asked the system to learn the 20 most relevant data patterns in this huge data set. And so then you come up with a vocabulary of biomarkers that are completely unbiased, because we as humans never told the system to find a specific biomarker, but the system just finds the most relevant markers in the data. And these markers include some examples that we are already aware of, such as subretinal fluid or here intraretinal fluid, but also new markers that we are not really sure what they are, but they, for instance, have a high correlation with visual acuity. So they seem to be relevant clinically as well. Now, in terms of practical disease management, it will obviously be very useful to have prognostic AI models that, for instance, can tell us the injection demand of a patient under intravitreal therapy. And also here, some proof of principle papers have been published that show that prediction of therapeutic requirements, for instance, and of treatment intervals is feasible with an accuracy that is maybe at the moment lower than what you would use in clinical practice. But these systems are still better than humans at the moment. And so I think in the future, perhaps we're able to get the accuracy of these systems higher up. And then these systems may also be used in practice to guide therapy and counsel patients. So to summarize really quickly, the current status of AI in retina, I think, is focusing mostly on replicating labor-intensive but potentially easy tasks that we do, such as screening or quantifying markers. And in the future, we will move to tasks that are more difficult, but that have a great potential to help us in our practice, such as understanding the link between morphology and function, finding new patterns and clusters in large data sets, and in the end, predict future outcomes and disease trajectories. Thank you very much for your interest.
Well, hello, I'm Tiernan Keenan from the National Eye Institute, and I'm delighted to present some of our work on using AI for some of these AMD tasks. The only potential conflict of interest to mention I've noted here. So what I thought I'd do is talk first about how we've trained deep learning algorithms to perform AMD detection and severity classification, so kind of at a single point in time. And then I'll move on to talk a little bit about our work using deep learning to try and look into the future in the form of personalized medicine in AMD. Maybe a very brief word about why we might be interested at all in training AI algorithms to do these kind of tasks. And I think, accuracy is one part of it, but probably only a small part. Actually, I think a much bigger part is massively expanding access to care. If we have safe and efficient algorithms we can deploy in primary care, optometry, etc., we can reach many, many more thousands or millions of people. Because, of course, there's limited retinal specialists and many hundreds of thousands of people walking around with early or even intermediate AMD undiagnosed. So to talk about the severity classification work, we call this work Deep CNet. Essentially, we were interested in seeing if we could train deep learning algorithms to do the same kind of detection and severity classification that retinal specialists might do in clinic. So essentially, for example, according to the ARED simple scale that we use a lot and its constituent features. So in this work, essentially, we were lucky to have many thousands of color fundus photographs at our disposal that were obtained through the AREDs. And of course, every single one of these has really detailed reading center grading for severity and all the constituent features. And of course, we use that for the ground truth. So in the usual way, we used many thousands of images for training, but we held some back for testing so we can report the performance of the algorithm. And actually, we can also compare AI performance with retinal specialist performance because we had the same images graded by 88 retinal specialists. So this is how we approach the problem. Rather than kind of end-to-end training where we give the images and ask the algorithm to do ARED simple score grading in one go, we actually broke the problem up. We had separate convolutional neural networks, one trained for drusen size, one to grade pigment, one for late AMD, etc. And that way we have much easier error analysis, more transparency. And so here's the performance of those three separate neural networks on each of those features. Essentially, you see really high AUC values, not far off 1, also represented by the blue curves. The equivalent performance of the retinal specialists on the same test set are shown by the red dots. So essentially, in most cases, we have the deep learning algorithms substantially outperforming retinal specialists on the same test set. So if they can grade these features, unsurprisingly, when those features are fed into the ARED simple scale, they can perform very accurate grading. And again, they substantially outperformed retinal specialists on the same test set. We performed training and testing for a lot of other, for example, the 9-step scale, geographic atrophy, reticular pseudodrusen. I'm not going to talk about that too much, or the external validation. I'm going to move on to talk about our work using deep learning to try and look into the future for AMD, trying to do kind of personalized risk prediction. And again, we were interested in seeing if we could train algorithms to receive a pair of fundus photographs from a single individual at a single time point, and just from that alone, generate percentage risk prediction. So how likely is this individual to develop late AMD over 1 year, 2 years, 5 years, 10 years, etc. And again, it's really important to compare that with our existing standards of care. So for risk prediction, that's of course the ARED simple scale for a simple version, or the KC calculator for a more complex version. So we essentially recapitulated those in the hands of retinal specialists so we could compare performance directly. I won't dwell on this too much, but essentially the ARED simple scale is helpful in the clinic, but it has a lot of downsides. It's fairly crude. The KC calculator overcomes some of these, but requires quite a lot more work on the part of the retinal specialist. And obviously, both approaches are totally limited by access to retinal specialist and to the accuracy of that specialist in being able to do the, quite difficult drusen size grading and pigment grading, etc. So, of course, for that reason, great if we can train AI algorithms that can do this safely, efficiently, with external validation, etc. And here's how we approached the problem. Again, we actually broke it up into 2 pieces. So in the first part, we had neural networks, a bit like the ones I described earlier, doing drusen pigment feature grading. And it's those features that are fed into the survival model that generates the risk predictions. In a second separate iteration that we call deep feature extraction, we actually allow the neural networks to choose for themselves which features they thought were most predictive. There can be many more of them, and those are the ones fed into the survival model. On the upside, we might get slightly improved predictive accuracy. On the downside, we have slightly more of a black box kind of problem. And essentially, to do the training and testing, we were able to combine ARIDS1 and ARIDS2 for a very large and a very challenging data set. So essentially, many progression events in different orders during training, which is great, but also many progression events during the test set, which makes a really challenging set. Essentially, the algorithm has to work really hard to discriminate who's going to progress and in which order. So here are some of the results. Essentially, on the top two rows, we've got the two existing clinical standards of care in the hands of retinal specialists. And for the same test set, on the bottom two rows, we have our two totally automated approaches. And the numbers shown are the C-statistic data. So essentially, the C-statistic is a bit like the AUC value for predictions. C-statistic of 100 means the algorithm's predictions lined up absolutely perfectly with the order in which people actually did progress, whereas 50 would be no better than chance. And essentially, you see the two totally automated approaches not only met, but actually substantially surpassed the predictive accuracy of our two clinical standards of care. Those were 5-year predictions, but actually we tested at every single interval, predictions at 1 year, 2 years, 5, 10, and the pattern was always the same. We can also see what are the predictions the AI algorithm makes for GA versus NV separately like. Essentially, they were both really good, but the predictions for progression to GA were particularly good, and I think that's probably helpful for a lot of the clinical trials we're interested in presently. We put all of the work, the detection, severity classification, risk prediction together in one desktop application, but I don't think I've got time to show you that, unfortunately, and the external validation work. So just to conclude, essentially, we can train algorithms given enough images and a good underlying ground truth to perform detection, severity classification, and even risk prediction very accurately in a way that outsurpasses our existing standards of care. I think this fits in well into this kind of cartoon, which shows a more wide context for what AMD tasks AI can help us with. All of the models and the underlying code, everything I've mentioned so far, is freely available for download to the community at GitHub. So if you search GitHub DeepSeaNet, you'll find it. It's for research use only. And I've also listed here some of the references, including the ones I've talked about, but also many that I haven't had time to talk about. I'm really grateful to our collaborators. This work was possible through an NIH collaboration through myself and others at the National Eye Institute with our computer science colleagues at the National Library of Medicine. So I'm really grateful to them and to our study participants. And many thanks indeed for your attention.
Hello, everybody. My name is Dr. Paisan from Bangkok, Thailand. It's my pleasure and honor to be invited to give this talk, Deep Learning, to detect OCT-derived DME from retinal photos multi-center study in the WOC of 2022. And here's my financial disclosure. So here's a conventional way of using deep learning for detecting diabetic macular edema from retinal photographs. We can train the algorithm using labels on the photographs based on the presence of hard exudates in the macula to classify the photos as having diabetic macular edema or non-diabetic macular edema. However, we often encounter cases like this, where cases with hard exudates at the macula on retinal photographs, but corresponded OCT did not show center-involved diabetic macular edema. On the other hand, cases with hard exudates at the macula, but OCT may show center-involved diabetic macular edema, just like in this case. There's an evidence in the literature showing that the prevalence of diabetic macular edema, if we detect based on the interpretation of retinal photographs, is different from the prevalence that we detect from grading OCT images. This paper was from JAMA Ophthalmology in 2016. Now let's take a look at this figure, where the upper part shows conventional or typical deep learning model for detecting diabetic macular edema from color retinal photographs, which was trained by data from color retinal photographs only. To better detect diabetic macular edema from color retinal photographs. Let's take a look at the lower part. We propose training from data on presence or absence of center-involved diabetic macular edema from OCT images by interpretation by either experts or automated annotations, and transfer the label data into the color retinal photographs. Here's our first publication on this approach, published in Nature Communications Journal in 2018. We used datasets from a retinal center in Thailand for this study. This model was initially developed by datasets from Thailand, as I mentioned before. 6,000 images for development and 1,000 images from internal validation, and external validation was performed on datasets of another 1,000 images from IPAX dataset. From internal validation of 1,000 color retinal photographs in the same dataset, you can see the blue line, which indicates ROC curves of the model, which perform better by the 3 retinal specialists who create color retinal photographs to detect DME. You can see the dots of the retinal specialists over here, but the line ROC curve is far better. As you can see, positive predictive value from the model was 61%, not high, but from the retinal specialist was around 37%. The negative predictive value from the model was 93%. The retinal specialist was 88%. Sensitivity from the model and the retinal specialist was about the same at 85%, but specificity was higher from the model at 80% compared to 48% from the retinal specialist. And this was validation of the new model of from the new data set of around 1,000 color retinal photographs. The model still performed better than graders from IPAX. Look at the blue line, which is ROC curve, and the red dot, which is from graders from IPAX. Here's a recent publication study of our model, published in Ophthalmology Retina early this year. The model was validated in multi-center, multinational datasets. Here are the baseline characteristics of the datasets from Australia, India, Thailand, and UK, with more than 3,000 eyes in total for the validation. As you can see, the ROC curves of the new model, which was higher than the ROC curves of the conventional model in each dataset from each country, and also higher in the combined dataset in the figure A on the upper left hand. For this picture, the gray column on your left hand over here indicated the prevalence of diabetic retinopathy and diabetic macular edema detected by conventional color retinal photo model. The gray column on your right hand is the prevalence of DR and DME detected by the new OCT-derived model. The middle gray column is the prevalence detected by adjudication crowd truth. You can see the brown stripe on the left, which indicates false positive cases of DME from the conventional model, which is bigger compared to the red stripe on your right hand, which indicated false positive DME from the OCT-derived model. This means the new model decreases false positive of DME significantly. So in summary, there are unmet needs for detecting DME from retinal photographs, since the indications of treatment DME are from OCT images. OCT devices are not for screening of DME in general populations like conventional retinal cameras. Our algorithm allows better detection of DME from retinal photographs. The validation of the OCT-derived models conducted in multi-center, multinational datasets still show better performance compared with conventional model. Prospective study on the algorithm and real-world deployment are on the way. I'd like to thank all the team with this study. Thank you very much for your kind attention.
Well, thank you to the organizers for the opportunity to present here today. I'll be talking about deep learning for the diagnosis of stage in retinopathy prematurity, accuracy and generalizability across populations and cameras. My name is Pete Campbell, and I'm from Portland, Oregon in the United States. These are my relevant disclosures. I don't have any relevant financial disclosures for this presentation, although I am broadly interested in artificial intelligence and ROP. What I'd like to cover today is the purpose, methods, results, and conclusions for this paper. The purpose of this study was to implement a convolutional neural network, or CNN, for binary detection of stages 1, 2, or 3 in retinopathy prematurity, and to evaluate its generalizability across different populations and camera systems. Why are we interested in this? Well, we've learned over the past 5 to 10 years that artificial intelligence can automatically detect ROP in images. That is, using digital fundus photography, you can automatically classify the zone, the stage, or plus disease in retinopathy prematurity. This review article that came out last year nicely summarized a lot of the different approaches, and I'd refer you there to sort of a good summary of the field. Why are we interested in this more broadly? Well, ROP is a big problem around the world. In fact, roughly 50,000 babies a year go blind or become severely visually impaired, primarily due to lack of screening. And that's because the population at risk is the largest in low-income countries, where the number of ophthalmologists available to screen is lower than it is in other regions. If artificial intelligence could be leveraged to help improve the efficiency and effectiveness of ROP screening, this number could be reduced. However, we know that artificial intelligence, when trained on certain populations, can be brittle, and they may not generalize. That is, they may not generalize to other cameras, they may not generalize to other populations, they may not generalize to other phenotypes. As an illustrative example, here's a figure on the right from a paper at the bottom showing images from the RetCam camera at the bottom and images from the Forest camera at the top. The Forest camera is a digital fundus camera developed in India by the company called Forest, which is available at a lower price point and therefore much more affordable in many low-income country regions. However, algorithms trained on one camera system may or may not generalize and perform well on other camera systems. So that's the motivation for this work. What did we do? Well, we used 2 large data sets, more than 5,000 fundus images obtained from the RetCam in North America, and more than 5,000 fundus photographs obtained by the 3-Netra Forest camera from 4 hospitals in Nepal. The images were labeled based on the presence of stage by 1 to 3 expert graders. 3 separate CNNs were trained using 5-fold cross-validation. A model was trained on North American data alone, on Nepal data alone, and on a combined data set of North American and Nepalese data. The algorithms were each then tested on 2 separate held-out test sets, images from Nepal alone and images from North America alone. Images were pre-processed as shown here. They underwent a 3-stage pre-processing designed to enhance the recognition of stage in the images, enhance the contrast, essentially. Here's what we found. These are, first of all, tables 1 and table 2, the trained splits and test sets of the different data sets. Images were all separated at the patient level. That is, there were no patients included in any of the separate splits and no patients included between training and testing sets. What did we find? Well, we found that CNNs can be trained to detect stage on either RetCam or Forest images and perform well when tested in distribution. That is, when a model was trained on North American data and tested on North American data, it performed well with an area under the ROC curve of 0.99. When a model was trained on Nepalese data and tested on Nepalese data, it performed well with an area under ROC curve of 0.97. However, when models trained in one population were tested in another population, the performance dropped. This is most notable when you look at the area under the precision recall curve and the sensitivity specificity at a given cut points. These dramatically decreased and to the point where they would not be effective for AI-based ROP screening. However, when you trained on images from both data sets, the performance in both test sets improved. That is, the more diverse and heterogeneous the data was on training, the better able to generalize externally the models were. This is a uniform manifold approximation and projection map, or UMAP, showing feature maps of the images in the data sets across both no stage and stage images and across the 2 data sets. What this shows is that even though model trained on both data sets was able to distinguish no stage and stage effectively in both data sets, it also was able to learn which data sets those images came from. This may be important when we implement and translate these models into practice around the world. In conclusion, artificial intelligence-based screening for the diagnosis of stage has the potential to improve the accessibility of expert-level diagnosis for multiple diseases. As we move from proof of principle efficacy studies, as shown here, to real-world effectiveness studies, a need exists to develop and validate generalizable models to different populations and camera systems. Put another way, we've seen in artificial intelligence and ophthalmology a lot of papers being published. We've seen far fewer models and algorithms implemented in practice, and there are several reasons for this. The motivation for this paper is one of those reasons. That is, sometimes algorithms developed in training and developed in silico don't work as well in the real world, and there could be many reasons for this. Heterogeneity and variability across populations and across different camera or device systems is one of those reasons. This paper demonstrates that training on more diverse and heterogeneous data set with respect to the intended use population not only improves external generalizability, but actually improved internal performance as well. Finally, overall, in order for these systems to be effective, we need to develop systems of care that integrate and sustain these technologies in health systems that lead to improved patient care and, in the case of ROP, more efficient, effective, and accessible ROP screening. Thank you so much for the opportunity to be here. I'd like to acknowledge Jimmy Chen, who is the first author on this paper, who's now a resident at University of California in San Diego and a really rising superstar. Thank you so much.
I thank all of you for the great presentations for this the AI in retinal disease, the 2022. Well, Peter, well, what do you think about the future of your model for ROP screening? The next step should be like real-world deployment. What's your plan for the future?
Yeah, great question, Paisan. So the paper I presented showed a model that detected simply the presence of stage in an image. And that would be analogous to a model detecting a hemorrhage in a diabetic population. And so I think it's really important as we think through which models get implemented in practice, asking the right question. And in my mind, the paper that I presented here was more of a proof of principle paper on heterogeneity in data and the importance of training on diverse populations and the effect of generalizability, and not that this is the right solution for ROP. And the reason is that in a typical population, roughly more than half the babies will have some stage, and you don't want half the babies to screen positive if this were implemented in screening. And so I think one of the questions we think through is how algorithms get implemented in practice, and do they answer an important question in the care pathway. And as Sebastian mentioned, detecting stage is not a complicated task.
Right.
And so I think we need to think through, how this gets implemented in practice. And for me, simply having a stage detector is not the most useful screening tool, because again, you'd have too many positives to make it useful. But I do think, and a lot of our other work has focused on more effective approaches to ROP screening using AI.
Yeah, well, Dr. Tiernan, probably the same question to you. Well, about the real-world deployment of the algorithm. I think that's the most important step. Using AI in healthcare, not only retinal disease.
Yeah, absolutely. And no, it's a big question. The kind of pathway from the proof of concept, right to clinical deployment is a long one, and probably may involve multiple different individuals, groups, stakeholders. So kind of academics probably can do some of those, but perhaps not all of them. Perhaps in some models it's important to pair with industry, who can take some of those steps more efficiently. So yeah, again, I probably would echo some of the points that we heard about in the last answer. We're still at an early stage in some ways. A lot of our work's a proof of concept. Right. If you see, the number of papers compared to the number of FDA-approved products, obviously that's a very high ratio. So in retina, for example, I think, like in AMD, which I do a lot of work on, I think it's important to kind of think early on about the use case, the setting and context. Because I tried to capture a little bit, but there wasn't much time in that kind of cartoon, which members of the collaborative community in AI published recently. What you might want for screening in optometry or primary care would be very different to what a retinal specialist might be interested in. The modalities may be different, detection, severity classification, retinal fluid quantification, so very different. So yeah, there's a lot in it there. I mean, I guess we personally have taken for some of our work, a different approach where we've recognized it's more proof of concept. We have done external validation, but we've made the code and models freely available to the community, recognizing that as members of the federal government, we may not have all of the parts to take it right to deployment. But yeah, I think maybe that answers some of the question. We can delve into bits of those in more detail if necessary.
Yeah, I agree. This is a very big question for us.
Yeah, but a very important one. Yeah.
How about we throw it back at you, Dr. Paisan? Where are we in terms of implementing diabetic retinopathy screening in Bangkok?
We want the whole country. So, well, in Thailand, we have the government-based support retinopathy screening for the whole country. So I think next month we're gonna doing a kind of, we call it implementation research. We're gonna use it in all the health regions in Thailand at the same time. We probably get approval from the Thai FDA in the middle of next year, and we're gonna deploy it for the whole country, maybe probably the middle of next year. That's our plan. I think for that, the retinopathy screening is it's probably the easiest among all those like algorithm for retinal.
Yeah.
Yeah.
I think I agree with that in some ways. The diabetic retinopathy screening, at least in some countries, particularly ones with quite a homogeneous health infrastructure, and I'm thinking of like my background in the UK, for example, it does feel like it may be an easier test case early on. Depending on the country and the disease, other places you might have to do more work to create the infrastructure. But from my understanding, Singapore, UK, those, perhaps Thailand, maybe it works more easily to use that with the existing infrastructure.
Yeah, same health system, right? We have kind of universal coverage from the
exactly, kind of a little homogeneous,
that sounds nice.
A long history of diabetic retinopathy grading centers, etc. Yeah, that infrastructure probably works well.
Yeah. So there's a question from the audience asking FDA approved AI system in ophthalmology. I think Dr. Tiernan replied already in the Q&A box here. Right. Do you want to?
Yeah, I was just having a quick look online. I believe the FDA keep a kind of continuously updated list. So obviously that's not specific to ophthalmology, but ophthalmology, is certainly amongst them, as we heard, was one of the first to receive approval. I think the majority are on the diabetic retinopathy side, but maybe not totally exclusively. So yeah, that's the FDA. I believe there are some other algorithms maybe approved in other regulatory domains, not necessarily by the FDA, so those wouldn't be included.
And I think Sebastian made a good point in his presentation that a lot of the ways AI may impact our clinical care is sort of behind the scenes in terms of the biomarker quantification. It'll end up on our machines and won't necessarily generate headlines like digital diagnostics does.
Yeah, I agree with you totally. Well, we may have some model for using for patients, but some model may be used for us, for the retinal specialists, to have a look at the kind of like tiny biomarkers that we cannot really detect in our clinical practice.
I agree. There's probably a bit of a distinction. Oh, sorry. I was just going to kind of distinction between ones that kind of really grab the headlines and we hope are going to be something big in the future, versus the kind of ones that actually probably save a massive amount of time and are not so kind of dramatically exciting. At our reading center, we talk about just really simple algorithms that can receive a whole ton of images, and they can, like 7 fields for diabetic retinopathy. They can do automated sorting, labeling, annotation, left, right, just very kind of simple stuff that's not super exciting, but can actually save a massive amount of time in the reading center.
Yeah, Peter, regarding to the FDA approval issue, do you want to have your algorithm to be approved by FDA? Is it requirement in the US, right?
Yeah, so the FDA pathway has had to evolve over the years as more and more AI algorithms sort of approach them for approval. And so it gets regulated under the sort of what they call software as medical device pathway. And CCOI has done a nice summary of this pathway and some of the considerations for ophthalmic software as medical devices. I forget there's a paper last year called Foundational Considerations of AI Algorithms or something like that, which nicely summarizes. And a lot of it comes down to, as we alluded to earlier, really the indication for use. And that has to be very specific. You can't just get an algorithm approved to use in any context with any camera for any, just for quote unquote detection of diabetes is not specific enough. And so it really does get very, very specific in terms of who the user is, which the camera system is, what's the intended population. And there's a defined pathway for each of those. That is, you might get an algorithm approved to assist clinicians doing some task, and that does not mean that that algorithm can be used autonomously, like the IDDX and like INOC, for example. And so, in answer to your first question, we have no plans to seek approval for the stage detector. Although, in work I didn't talk about today, we are trying to do that for a sort of plus disease scale, which seems to be a more effective way to sort of diagnose ROP and screen for ROP.
I see. That question from the audience asking about I'm going to read the question. Do you guys think OCT made in centers, probably retinal centers, could play a major role in screening diabetic retinopathy patients and sending data to cloud? So I think they asked about the using OCT, based OCT analysis for screening. Tiernan, you want to address this?
I can give you. So if I understand it correctly, it's talking about OCTs obtained in a retinal clinic. Then would that be?
Yeah. You can read that in the Q&A box as well. Yeah.
Well, I guess so.
But your work is about color fundus photos, right? It's not.
Yeah.
Actually, Sebastian should be the one who address this.
Yeah. I mean, there's could be a lot of questions there. There's the question of what modality. So both the modality and the location. So if you're, in primary care, maybe it's more often going to be color fundus photographs. If you're in, ophthalmology, whether it's general, comprehensive, or retinal, OCT is going to be more readily available. I think the cloud-based question is a good one. I have some experience with the kind of a paradigm. Let's say, Notal, not only Notal, but others as well. But Notal are, a decent way along with this. The idea, like an individual with neovascular AMD has a home OCT device, daily or regular image acquisition just by the patient, him or herself. And then that's sent, electronically to the company's cloud, and the AI algorithm is applied to that. And you can do automated retinal fluid detection and quantification. You can plot it on a graph and correlate it against your anti-VEGF injections. That's so the cloud-based approach, given sufficient security, I think is a very viable one. I think we'll see more of that in the future. I'm not sure how to answer the rest of the question about the OCT. I guess screening normally would be not in the clinic. It would be more in primary care.
I kind of like this question because I've always thought that the first answer is obviously fundus cameras are cheaper than OCTs, and so that's why I think the first approach to screening has been with fundus photos. That said, OCTs, the pieces of the components are not that expensive, and you could build a low-cost OCT for $5,000 and put it in the clinic. So really, it's about two things. It's about and you'd have a device there that would be more sensitive and more specific for more diseases and things that we're learning about in terms of neurodegeneration is not endless. If we could give the two limiting factors, I think, are one, systems of care, because there's no financial model for that to be a profitable business and sustainable at the current point. And secondly, again, as I mentioned earlier, from a regulatory perspective, you have a challenge to get one device to do multiple things. And so you'd have to think through carefully what the purpose of the device is and what you're screening for, and you'd have to be validated for a lot of different diseases, many of which are low prevalence. So that's going to be challenging. But I love the idea.
Yeah. Yes. Well, because screening should be performed in the population with unmet need, right? In the rural areas. Well, if you have like a kind of small portable OCT, is kind of you can bring over to all those places. is going to be very useful instead of the retinal camera that we are using at the moment. It's going to be very, very useful. Yeah, agree with that. They look at it.
And I think the group in the UK has demonstrated, that OCT in sort of primary optometry clinics can be used to sort of screen for referral warranted retinal disease, for example. So in a more distributed model, not just in the retina clinic, you can use AI to sort of make the retina practice more efficient with those with higher acuity disease.
Yeah. The thing is, in the UK, you have like the quite many optometrists, right, practice, and they use OCT in their optometrist clinic. Mm-hmm. That's the reason why you need the OCT model in the UK, right?
Yeah. So not every optometry office in the in the UK would have an OCT, but many did, over the past few years, kind of put that money in up front. And like we were hearing about, often they've established referral pathways with local ophthalmologists, retinal specialists, where they securely send the data, to cut down on unnecessary patient attendance. They can do and not just unnecessary attendance, but a kind of prioritization, a stratification of the referral. So, obviously looking out for neovascular AMD, which needs to be seen very quickly, but then diabetic macular edema can be seen at the next level of urgency, and so on. So I think that has been very successful in the UK. But not every optometrist has an OCT. It's not like a government implementation. It's a private decision.
Yeah. Thank you for your answer. But I think the first presentation today, it's night time here. Sorry. Well, I think it's quite interesting of using the ultra wide field for screening diabetic retinopathy, because we can see a lot at the periphery. What do you think about this? Could this become like the major change in the future, or what do you think about it?
I think my answer would be the same, which is that it's about systems of care. So if you can get that, into a primary touch point where patients can be screened, and that's effective at the population level, I see it as a similar approach to IDDX in terms of, you could use it autonomously, or you could do a telemedicine reading. It's definitely a useful camera. I think it's just a matter of getting a system that makes that sustainable, because the camera itself is not that inexpensive.
Yeah, I see. But actually, the ultra wide field camera at the moment is in commercial, right?
Mm-hmm. I agree, and I mean, it's very attractive because potentially, such a wide variety of pathology from the macular periphery that one will be able to capture in one go. Like in diabetic retinopathy specifically, which I guess the first presentation was on, we're learning more and more about predominantly peripheral disease and so on, and probably more to learn there still about the role that kind of disease plays. But I agree that probably the fundamental question will be the organization of the care structure, the economics behind it, and so on. But technologically, it should be possible for sure.
Yeah. Well, Tiernan, you said, well, you have the external validation in your AI algorithm. Right. Well, how big is your data set?
A lot of different projects, and we've done a lot, different data sets for external validation. Just, I guess I could give a couple of examples. Let's say, like in some of them where we've used, the primary data set for training and testing would be AREDS1, for example. Then we can perform external validation on the AREDS2 test set, which in a way is obviously it's limited to the US, and it has a very particular ethnic distribution, with very few non-Caucasian participants, but that's certainly a limitation. But it has the advantage that AREDS2 was taken from, 82 different clinics across the breadth of the United States on, so that means a ton of different cameras, many different photographers, etc. In other experiments, we've performed external validation on, data sets from Europe, for example, the Rotterdam eye study, which has been very helpful. We've done a very big cataract project. Yeah. We've done external validation on data sets from Singapore. So, but I think, and we've alluded to this, many speakers along the course today, the importance of that.
Do you think nowadays we still need quite a big data set for external validation, or?
Yeah, I personally think it usually doesn't have to be as big as the initial So I guess the main place where the number of images and the ground truth is really important is for the training purposes, where often, I mean, maybe you can do pre-training, but still you're going to need ideally hundreds or thousands of images. So I think the external validation data set doesn't usually have to be quite as big as that. It has to encompass the spectrum of disease you're interested in testing. It has to have great ground truth, obviously. But I think it's particularly training where you have to have that very large size, ideally.
Yeah.
On your work, so I've always wondered. It's fascinating that you can learn, better predictions from these fundus photos about progression to AMD. That seems to be really useful to retina specialists. But in my practice, I don't get a lot of fundus photos. And so one of my questions in terms of application is, and I'm sure you've thought of this, but I don't know if anything's been done. Have you tried to apply the models using transfer learning to on-phos OCT infrared images or some other, or try to do the same questions using OCT?
Absolutely, yeah, no, great question. We've done that in some circumstances. We've done a lot of work on reticular pseudodrusen where we've looked at every single different modality, transfer learning where the ground truth comes from the FAF or infrared image, but you're training and testing on the color fundus photograph. Together with some colleagues, Kathy Kukras and Thurindu De Silva, we've done work on geographic atrophy, including, like FAF versus on-phos OCT, that kind of thing. So yeah, I think those are great questions. I agree, depends on the device. I quite like some devices where you simultaneously get the OCT and the color fundus photograph, but it's true, often we're not getting them these days. Yeah, I guess that one advantage, at least in AMD, there's so much work on OCT, but still, our features on color fundus photographs are so well validated in terms of risk prediction, severity, and that's not quite there yet with OCT. I'm sure it will be in the future, but we've not had a kind of ARIDS 1 or ARIDS 2 with OCT features. I mean, hyperreflective foci, obviously there are correlations, but at least if you tag it back to the CFP features, then you've got something quite hard to correlate with. But I love the multimodality work. We've done a lot of that.
Yeah.
Well, and GA is another big question we've not got into, but that will be really important for the future, I think, for clinical trials right now, but being able to deploy AI devices, even in routine clinical practice, when we have the first approval of a drug to slow geographic atrophy enlargement, we're going to very rapidly need to know what is the GA area size right in front of me today? What was it 3 months ago? Has the enlargement slowed? Ideally, GA enlargement risk prediction. So that's soon going to become really important. And again, whether that's from OCTA or OCT or FAF, these are open questions.
Yeah, I think I have the last question, maybe for both of you, about the image standardization issue, right? Because, we didn't use DICOM as in other, we're using JPEG, we didn't use DICOM. Think about the imaging standardization. It's going to take like many years. Can we get there? What do you think about it?
I'll try and to give a short answer to that question, which is, I think there's a lot of talk about AI and big data and all of this, but a lot of the most important work that needs to be done is not sexy, and it's stuff like this, which is is getting cameras to talk to each other, and the power of our big data would be greatly amplified if we could solve that problem. And there's people working on it, and so the short answer is I hope so.
Yeah. Okay.
Yeah, and I'll keep that's a very brief as well, but there's a tremendous amount of effort and attention in this area from like NIH workshops, multiple editorials and publications. It's happened in cardiology and in orthopedics, other fields. The devices, the MR, the CT, they talk to each other. I don't know why it's been slower in ophthalmology, but there is definitely there has been a lot of success recently to bring industry, academic, other stakeholders together. So again, I hope so, and I think it probably will.
Well, I think our time's up. Well, thank you very much, Peter and Tianan, for your participate in this discussion. Thank you very much.
Thanks so much.
Thank you.
Yeah, and thank all the speakers who joined this session and thanks all the audience for joining us tonight. And maybe today, in the morning, maybe in the daytime. Well, thank you very much all. I declare this session closed. Thank you, Peter. Thank you, Tian.